Hello. So in this process, I'm going to explain the process of fermentation, why fermentation happens, where it happens, its comparison to cellular respiration, etc. So to recap cellular respiration on a high level, cellular respiration's goal is to produce ATP. And this whole process really just generates ATP. And for that to happen, we have four stages called glycolysis, oxidation of pyruvate, citric acid cycle, and oxidative phosphorylation. Something that's common to both process fermentation and cellular respiration is glycolysis. In glycolysis, we know that glucose gets converted to pyruvate molecules. In the whole process, we also generate a small amount of ATP called 2-ATP, and we also need NAD plus in glycolysis that gets converted to NADH. Now, in glycolysis, NAD plus plays a very crucial role. NAD plus is required for glycolysis to happen, which means for it to happen again and again and again, in the whole process, it needs to get regenerated. And we know that in cellular respiration, NAD plus gets regenerated because after all the really detailed and complex processes happen through glycolysis, oxidation of pyruvate, citric acid cycle, and oxidative phosphorylation, we know that a large amount of ATP is made, and we know that in the last stage, which is oxidative phosphorylation, once electrons kind of go through the electron transport chain, we have oxygen acting as the final electron acceptor in the end. And oxygen, once it accepts its electrons, NADH will get oxidized to NAD plus again, because NADH is the electron donor to the ETC, right? So in the whole process, we know that NADH will get oxidized to NAD plus, and again, NAD plus in glycolysis will help glucose conversion to pyruvate, and the whole process continues in cellular respiration. Now, when we look at fermentation or anaerobic respiration, as the name suggests, there is no oxygen, right? In fermentation, there's no oxygen. So who's the final electron acceptor? And what role does oxygen play? What role did oxygen play in cellular respiration? Because oxygen accepted the electrons, NADH could be converted to NAD plus again. In the case of fermentation, again, in fermentation also the goal is actually to produce ATP. In glycolysis, the goal is to produce ATP. Now, for producing ATP, we need NAD plus. How are we going to regenerate NAD plus? That is the goal of fermentation. That is what fermentation does. Regenerate NAD plus so that glycolysis can occur and ATP can be produced, right? So let's look at fermentation right now. Obviously, in the first step, we know that glucose is converted to pyruvate, and we have the reactants, which I will circle. So these guys are the reactants, and these guys are the products in glycolysis. So we obviously know that we have glucose. We also have 2-ADP and 2-IP generating 2-ATP. Note that there is actually 4-ATP that's generated in the process of the conversion of glucose to pyruvate, but I could be wrong over here, so just take this with a grain of salt. I know that in the whole process, 4-ATP is actually made, but 2-ATP get used up, so the net ATP gain is just 2-ATP. We also know that there is 2-NAD plus, which gets converted to 2-NADH, which is actually what is shown over here, but we'll get into that process a little in a bit. So, the glycolysis stage is common to both alcohol fermentation and as well as lactic acid fermentation. Okay, from now, we know that after glucose has been converted to 2 pyruvate molecules, Also note that in this process, NAD plus also has to gain electrons to get converted to NADH. We know that we always know that when something gains electrons, it gets reduced. To NADH is actually reduced to NADH. Now from here, let's see how alcohol fermentation happens. The 2-pyruvate molecules actually get converted to 2-acetaldehyde through a catalyst called pyruvate decarboxylase, and in the process 2 carbon dioxide molecules are eliminated and an intermediate product called 2-acetaldehyde is formed. Now at this stage, NADH will donate its electrons to 2-acetaldehyde, and obviously when something gains electrons, it will get reduced. So 2-acetaldehyde gets reduced to 2-ethanol. So when NADH donates electrons, it will get oxidized to NAD+. And remember when I said one of the main goals of fermentation is to actually regenerate NAD+, that is what is happening over here. By NADH losing its electrons, it's getting oxidized to NAD+, which will go over here and the process will continue. So this is alcohol fermentation. Now let's look at lactic acid fermentation. After the conversion of 2 pyruvate molecules, the pyruvate molecules will actually gain electrons from NADH. So NADH will donate its electrons to 2-pyruvate, and pyruvate molecules will get reduced to 2 lactate molecules. Now obviously again when NADH donates its electrons, it will get oxidized to NAD+, which is again the whole point of fermentation, so that glycolysis can occur and ATP can be produced. Now let's look at where these whole process happens, right? So we know that in cellular respiration, there's obviously a nice electron transport chain, and there's an oxygen sitting there conveniently to accept electrons and for NAD+ to be regenerated. In this, which means in fermentation, there's no electron transport chain. There's no certain membrane in which there are ETC in which there's an ETC sitting. There's an oxygen acceptor. There's nothing like that over here. So this actually happens in the cytosol, which is the cytoplasm. And alcohol fermentation mainly happens in yeast, bacteria, etc. And lactic acid fermentation actually happens in muscle cells. Like, for example, when we get cramps, that is an example of lactic acid fermentation, when a lot of energy is required but there's not enough oxygen. So the cell will quickly move from an aerobic respiration to anaerobic respiration. So that sums up anaerobic respiration or fermentation.